Dear students, in this module, I'm going to elaborate on another type of secondary structure in proteins, that is, the coil. You know that alpha helices and beta sheets are fixed secondary structures and are very important in the overall folding of the protein. Moreover, you know that loops are flexible secondary structures and are useful in connecting the alpha helices and beta sheets. Moreover, these loops, they constitute active sites in the proteins and are ports for interaction with other proteins. So, what about the coil? Coil is another form of secondary structure, and to identify the coil is a difficult task. The reason for that is that anything that is unstructured within the protein structure, we call it a coil. So essentially, any structural artifact, any structural form within the protein backbone that is not an alpha helix, that is not a beta sheet, that is not a loop, is classified as a coil. The lack of structure in the coil does not mean that it is not important. The coils have been shown to perform very interesting functions in the proteins. It will not be incorrect to say that our understanding of the coils or the unstructured regions within the protein structure is very limited. So, these coils are randomly oriented and they are connecting loops, beta sheets, alpha helices, and are there in the overall structure of the protein. I will show you an example here. So this is the myoglobin protein, and you can see here there are some unstructured regions within the protein backbone. So these regions are actually the coils. Also another one is here, and there are some portions within the protein where you can see these are the loops. So in this way, loops and coils, they come together and they organize the overall structure of the protein, in this case myoglobin. So in conclusion, coils are those secondary structures which are neither alpha helices nor beta sheets nor loops, but that they are unstructured and they may be long in length and that they may be performing several critical functions in the protein. As I mentioned earlier, our understanding of the coil structure is still limited and science is proceeding on to better understand the coils. Therefore, the coils do not have a consistent classifiable structure.